Mycobacteria are a group of bacteria, and there's lots of different types. Many of them are found in soil and water and are essentially harmless to most people. But there are a few mycobacteria that actually cause very devastating human diseases. And one of the most famous is Mycobacterium tuberculosis, which causes tuberculosis. There's another mycobacteria called Mycobacterium leprae that causes the disease leprosy. There are several unique features. First is that, different from other bacteria, it has a very unusual cell wall or cell envelope architecture. And the cell envelope is basically the thing that coats the bacterium and protects it and interfaces with the environment. Mycobacteria have a very unusual structure to its cell envelope. It's very thick, it's very waxy, and we think that that prevents a lot of antibiotics from actually getting into the bacterium to reach their target and to kill the bacterium. And then the second feature that we think is really unusual to mycobacteria is that even in a genetically identical population, mycobacteria seem very good at generating a lot of diversity in different physiological properties. We think that differences in these mycobacterial populations at the single cell level can be really important for the bacteria to tolerate antibiotics, meaning that there are some bacteria that can survive antibiotic pressure simply because they phenotypically look a little different than others. How are individual mycobacteria different? How do they differ in the responses to antibiotics? What are the underlying causes of those differences? We think a lot of it traces down to metabolism, and so my lab is really trying to understand the metabolism of individual mycobacteria. We use microscopes to answer those questions. That can range from simple time-lapse videos where we look at individual cells over time, and because some of them can grow and divide really slowly, we have to run our microscopes for up to 5 days in order to capture the cell growth and division of these organisms. But we also use super resolution microscopy to understand the spatial organization of proteins within the cell. We also started using these fluorescent reporters or sensors for different metabolic properties of the cell. For example, we have used a fluorescent reporter for ATP, which is the energy currency of the cell. And so now we can measure ATP levels in individual cells and understand how differences in say, ATP or other metabolites are generated in a clonal population and what are the consequences for having different levels of these metabolites. Our goals are treating some of these mycobacterial infections. Tuberculosis kills more people each year than any other single infectious disease, and in history has actually killed more people than any single infectious disease. So it's an incredible global health problem. If someone gets TB, they have to go on a course of antibiotic therapy that can last anywhere from 4 to 6 months with a combination of 4 antibiotics, which, as you can imagine, is a very long time to be on antibiotic therapy. We think that if there are all these differences in metabolism and physiology of that bacterial population, then we think that if we can transition cells or force cells into a more antibiotically favorable state, that we might be able to treat an infection more rapidly.